Assalamu alaikum wa rahmatullahi wa barakatuh. I'm teacher Ahmed Jamul. I'm glad to talk about document 4 of chapter 1, Spermatogenesis. I'm going to discuss the histology of the testis, describe the spermatogenesis mechanism, and end with a scheme about spermatozoon. As you have seen earlier in document 1, the male reproductive system is formed of the male gonad, the testis, which is the site of the sperm production. The genital tract that is responsible for both conduction and the storage of sperms, and that starts with the epididymis, followed by the vas deferens, then the urethra. And finally, the accessory glands that are responsible for the production of the seminal fluid. In document 3, you have learned some of the basics about the process of meiosis by which gametes are produced. In this document, you will get to learn more about the histology of the testis and how does the genesis or production of sperms occur within this testis by a process called spermatogenesis. What you see in this figure is a longitudinal section of the testis and the epididymis. The section shows that the testis is divided into smaller rooms called lobules. Within each of those lobules, you can see some tightly coiled structures called seminiferous tubules. If a cross-section is made through some of these seminiferous tubules to observe a top view under the microscope, you will observe something similar to document B of your books. So this document shows a cross-section through the seminiferous tubules. In this cross-section, you can detect the 3 seminiferous tubules. In between these tubules, you can observe some cells that are called Leydig cells or interstitial cells. These Leydig cells secrete the male sex hormone testosterone, which stimulates the production of spermatozoa. Now let's closely observe one of these seminiferous tubules cross-sections. It has a circular shape with a thick wall made up of many cells and a lumen in the center filled with sperms. There are different cells observed within the wall of the seminiferous tubule. The germ stem cells, the spermatogonia, are found at the basal lamina of this tubule. Then, as you move in a centripetal way towards the lumen, you will observe new cell types, spermatocytes, spermatids, until finally you will observe sperms which are released into the lumen of the seminiferous tubule. Document C of your books shows an illustrated view of the cross-section of a seminiferous tubule. Here you can clearly notice the centripetal direction of the sperm production, or what we will start calling spermatogenesis. In this document, you can also see that this tubule contains cells other than the germ-line cells. They are somatic diploid cells represented on the figure in yellow. These somatic cells are the Sertoli cells that surrounds and nourishes the germ-line cells during their different cell divisions. Now that you know that spermatogenesis occurs in the seminiferous tubules and that it is stimulated by the testosterone secreted by the Leydig cells in between those tubules, and that Sertoli cells nourishes the dividing cells during spermatogenesis, it is time to learn in details about what happens during spermatogenesis. The process of spermatogenesis starts at the basal lamina of the seminiferous tubule, where undifferentiated spermatogonia are located. Those germ stem cells continuously multiply by successive mitotic divisions that produce similar cells that stay at the lamina as well. This is the multiplication phase of the spermatogenesis. But starting from puberty, few spermatogonia periodically move away from the lamina towards the center and enter an interphase to become spermatocytes 1. This is the growth phase of spermatogenesis. You can see that the number of chromosomes in both spermatogonia and spermatocytes 1 is still the same, equals to n. But since during interphase a duplication of genetic material occurs, then chromosomes in spermatocytes will be duplicated, made up of 2 chromatids, each, unlike those in spermatogonia, made of individual chromatids. Each spermatocyte 1 undergoes the reductional division of meiosis to produce 2 spermatocytes 2 that have half the number of chromosomes N, but since only homologous chromosomes separate in this reductional division, the chromosomes in the spermatocyte 2 are still duplicated with 2 chromatids. Now, spermatocyte 2 in turn undergoes the equational division to produce 4 spermatids. In an equational division, there is no change in the number of chromosomes, so spermatids, just like spermatocytes 2, have N chromosomes, but in an equational division, the separation of the sister chromatids of a chromosome occur. That's why spermatids have chromosomes with individual chromatid only. The whole meiosis refers to the maturation phase of spermatogenesis. The last phase in spermatogenesis is the differentiation or spermiogenesis. By the end of this phase, you can see that the spermatids turn into cells with a different shape but the same number of chromosomes N. That's because spermiogenesis does not include any further reduction. It only includes morphological changes necessary for the function of the sperms produced and will be released in the lumen of the seminiferous tubule. Spermiogenesis includes the disposal of all the excess cytoplasm of the spermatid as shown in document E of your books. Only the most important organelles persist in a sperm cell: the acrosome that contains enzymes needed at fertilization, the mitochondria that are needed at the center to provide the sperm with the energy needed during its movement, and a flagellum appears to help the sperm with its propelling movement. And this is the mature sperm shown in document F of your books, would be the final result of spermatogenesis, formed of 3 parts: the head, the midpiece, and the flagellum. To wrap up, you learned that the testicle is made of many seminiferous tubules. Spermatogenesis occurs within those tubules in a centripetal way in 4 phases: multiplication, growth, maturation, and finally spermiogenesis, by the end of which a mature sperm is produced. Hope this video was helpful. Please read the corresponding part of the summing up